Okay, good afternoon, everyone. Hope everybody had a good week and a good weekend. So today, a couple things coming up. Today, I'm going to be doing Enterobacteriaceae, the second lecture, and actually, I want to try to get the third Enterobacteriaceae lecture in today. And next Wednesday, I'm not going to be able to have a Zoom lecture with you. I'm going to be out of town next Tuesday and Wednesday. But what I'm going to do is, since these are Zoom lectures anyway, over the weekend, I'm going to uh, do the Zoom lecture for next Wednesday, and I'll send that to you. And then when I'm back on Thursday, then we'll go over it. We'll quickly go over that lecture and go over any kind of questions that you may have on that lecture. The reason why I want to do two lectures today is that I kind of noticed a little bit on the quizzes that there's some points that maybe it's my fault, but I'm assuming a lot, and maybe I'm not driving points home clear enough. So tomorrow, instead of having a lecture, and that's why I want to do two lectures today, I want to do review. You got three quizzes on Friday, and then you have your first exam number one. Exam number one, it shouldn't be too bad. Of course, it's easy for me to say because I made the exams. So so I'm going to try to do two lectures today, and I'm going to try to make it as simple as possible, easy to understand. And the points that I really want you to know, like for example, the indole test. The indole test, you're testing for tryptophan. All the other information about the indole test is I want to really make it secondary. So whenever you get a test question that has to do with indole and tryptophan, those two terms go together. So as I go through these lectures, I'm going to really emphasize that this is what you need to know, and I'm gonna really highlight those points. So I'm gonna go ahead and start with this lecture, lecture 7, Enterobacteriaceae 2, and then after that I'll do Enterobacteriaceae 3. And then next week's lecture, well, we'll talk about it tomorrow in class. We'll have plenty of time because I won't be lecturing tomorrow. We'll be doing a review, and we'll also be doing Kirby-Bauer sensitivities in the laboratory. Okay, Enterobacteriaceae, lecture 2. So you're going to be able to identify, and that's the main thing, identify. So in this class, especially for microbiology, the key thing is to identify the organisms. We're going to go through a lot of biochemicals that will be, as you've noticed in the staff and the strips, there's biochemical tests that are performed, specific biochemical tests that will be positive for specific organisms. And that'll be part of the review tomorrow, and I'll point that out. Okay, so urease, the urease test is testing for the organism if it has the urease enzyme. That's what we're testing for, the presence of the urease enzyme. And in order to do that, we introduce a medium, or a substrate, if you will, to the organism. And if the substrate is broken down by the organism, then that means the organism has the enzyme urease. There's a little, and then certain organism has this enzyme. So it's a pretty simple test. We use Christensen's urea agar. It's a solid agar and it's a slant. There's carbohydrates, and of course it has to have urea. That's the main thing, main substrate that we're looking for. And then there's a phenol indicator. So to do the test, you inoculate the streak. It's pretty simple. Just inoculate the streak, incubate it overnight for at 37 degrees, 35 degrees. And the next day, all you do is look for a pretty pink color if it's positive, and it's yellow to light orange if it's negative. Quality control is Proteus vulgaris. That organism is positive, and E. coli is negative for urease. So those two organisms are used for quality control. You can use other organisms too, like for example, uh, or Enterobacter or other organisms, Serratia, but E. coli, it's pretty safe to say that it's negative for urea. Proteus vulgaris is positive. And then also too, since you're going to be having your Enterobacteriaceae exam, the flow charts. I'm going to go over the flow charts next week for the review, and because you're going to be getting your Enterobacteriaceae exam coming up. Okay, and I'll go over the Ewing groups. So there's urea slant on the left, uninoculated, and urea positive. It's nice, pretty pink for positive, and this is the urease negative. The next one is decarboxylase. So, and it involves three different amino acids: lysine, arginine, and ornithine, and each of those will have a specific end product. So we're testing for the organism's ability to decarboxylate certain amino acids. Okay, so lysine decarboxylate, the amine is cadaverine. This you have to memorize. This is definitely going to be on the test. So lysine decarboxylase, LDC, the amine is cadaverine. Ornithine decarboxylase, the amine is putrescine. And arginine decarboxylase, the amine is citrulline. So make sure you memorize which goes with which particular decarboxylase enzyme. These are the enzyme substrates, and these are the amines. Okay. And they may be performed separately or in combination media. This is another thing that's going to be, that you're going to be asked. It's going to be on a test. What are the conditions for decarboxylation to occur? The environment has to be acidic, low pH, low pH. The conditions have to be anaerobic, and that's why we do an oil overlay. We just put a few drops on top of the substrate and overlay it with oil. What that does, it creates an anaerobic condition. And it also has to possess, obviously, the appropriate enzyme. The arginine decarboxylase, lysine decarboxylase, and ornithine decarboxylase. Those are the specific appropriate enzymes. Okay, so those three things need to be present for the decarboxylase test to occur. When amino acid is decarboxylated, it reverts to its original color. Okay, so combination tubes. There's a tube called the MIO, which stands for Motility-Indole-Ornithine. MIO, Motility-Indole-Ornithine, which means on this tube, this is one tube where you can perform three tests: the motility test, the indole test, and the ornithine decarboxylase test. The LIA is another combination tube that we're not going to use. It's lysine iron agar. But on the MIO tube, okay, going back to, these are the amino acids: lysine, ornithine, and arginine. The tubes consist of 0.5% glucose. There's peptone and enrichment, and the pH indicators rum cresol purple or cresol red. Like I said, you want to overlay the tube with mineral oil to create anaerobic conditions. Remember, it's one of those three, and the oil creates anaerobic conditions. Incubated at 37 degrees overnight. Okay, 18 to 24 is pretty much overnight. And this is one of the reasons why I don't like this test, because a positive test in this tube, and we're going to be getting these tubes too, is and so that we can actually look at the reactions, is that a positive test is purple and turbid. Okay, negative test is yellow, but and also too, a negative test is purple and clear. So there's a difference between purple and turbid and purple and clear. Okay, yellow is also interpreted as being negative, but if it's purple, but if it's clear, then that's considered a negative test. It has to be turbid to be called positive. This is another test. Make sure when I say that these are going to be on the exam, you're putting asterisks or you're marking the pages, or you're putting a big note. Memorize this, okay? These reactions with these organisms, lysine decarboxylase, ornithine decarboxylase, and arginine decarboxylase, it's positive and negative for these organisms. This is how to distinguish between Kleb pneumo and Enterobacter cloacae. It's pretty easy. If you set this up, you notice that Kleb pneumo is positive here, and the other two are cloacae. And for the negative, E. cloacae is negative here and positive for Kleb pneumo. So make sure that you know how to set up your table and memorize it. But it's going to be, this table is going to definitely be on either a quiz or a test. Okay, indole testing. This is the one I mentioned earlier. Indole is the end product of the deamination and hydrolysis of the amino acid tryptophan. Okay, so associate indole with tryptophan. The media that you can perform the indole test on is blood agar plate, the blood agar plate, the tryptase, the TSA, tryptase soy agar, chocolate SIM agar, which I'm not familiar with, don't use it. Indole nitrate, which I don't use, and the MIO. The MIO I just mentioned, which is the Motility-Indole-Ornithine, but not McConkey. You don't want to do an indole test on, excuse me, on McConkey, because McConkey is pink, okay? And that's the reaction that we are looking for, a pink or reddish color. And if you're scooping up media or the agar, then you're going to mistake that as being positive. So the indole test is a rapid screening, and you usually do it on blood. Two types of indole test is the dimethylaminocinnamaldehyde, DMACA, and the Kovacs reagent is dimethylamino-benzaldehyde. The cinnamaldehyde reagent is used for taking direct colonies and doing the indole test off a direct colony. You either use a filter paper or you can use a swab. The benzaldehyde, dimethylamino-benzaldehyde, is if you're testing it on liquid, a tube method, like for example on the MIO, remember the Motility-Indole-Ornithine, well, you can do the indole test using the Kovacs reagent. And the Urease test is another indole reagent, but we won't be using that. The rapid test, like I said, take a cotton swab or smear colony on filter paper and 2 to 3 drops of reagent of the dimethylamino-benzaldehyde. What we have are pinch vials, these little vials. You pinch it and it releases the reagent, and then you squeeze drops out of it. So that's the indole test. And then what you do is you look for a blue color. Okay. Indole test Kovacs, that's the dimethylaminocinnamaldehyde. You're going to take 0.5 mL of the Kovacs reagent, put it on top of the MIO tube, and look for a pink colored ring at the top of the tube. And this is the MIO. As you can see, there's a stab there, and you're looking for motility. This is indole negative, and this is actually positive indole. It's also positive for motility. See how the tube below the pink is pretty diffuse? That's positive motility. Here, around the stab, not much motility took place, but here, a lot of motility throughout the tube. So this is positive motility, and the pink indicates positive indole, okay? Controls, indole positive is E. coli, negative control is Kleb pneumo. So make sure you know that what we'll be studying are in the Enterobacteriaceae, there are 3 organisms that are positive for indole. One of them is E. coli. The second one is Kleb pneumo, and I'll talk about how to distinguish, Kleb oxytoca. I'll show you how to distinguish between Kleb pneumo and Kleb oxytoca. Oxytoca is indole positive. But morphologically, on the colonies, they look exactly the same. Okay, so but the only difference is that Oxytoca is indole positive. Also, two Proteus. There's Proteus vulgaris and Proteus mirabilis. Both of those organisms swarm the same way, but you tell the difference by doing an indole test. Proteus vulgaris is indole positive and mirabilis is negative. So those are the three organisms that test positive for indole: E. coli, Proteus vulgaris, and Kleb oxytoca. Okay, oxidase. For Enterobacteriaceae, all Enterobacteriaceae are oxidase negative. Enterobacteriaceae are oxidase positive. This test will test for the presence of the organism having cytochrome oxidase, and the reagent is tetrapyraphenylene diamine dihydrochloride, forming indophenol, which is a blue to purple color. But the reagent is I'm not gonna make you repeat it, say it, or spell it out. But it's tetrapyraphenylene diamine dihydrochloride. So, like I said, all Enterobacteriaceae are negative. And the organisms, and we're going to be studying them coming up, are oxidase positive, like Aeromonas, Pseudomonas. Now, anything that has a monas at the end of it, like Aeromonas, Pseudomonas, Plesiomonas, etc., anything those are called the monads. The monads are oxidase positive, and they have polar flagella. Okay, Neisseria. So the flagella characteristic doesn't apply to Neisseria because Neisseria is not motile, but Neisseria is oxidase positive, Campylobacter is oxidase positive, and Pastorella. We're going to get into Pastorella pretty soon with the aerobic GNRs. But all of these organisms, the monads, the Aeromonas, Plesiomonas, Pseudomonas, they're oxidase positive. These are oxidase positive organisms. But for Enterobacteriaceae, for your first gram-negative GNR exam, all are oxidase negative. Okay. Remember your characteristics of Enterobacteriaceae. Like I said, the reagent is the tetrapyraphenylene diamine dihydrochloride. That's Kovacs oxidase. There's also a Kovacs indole. Remember the Kovacs indole, which is the liquid? Well, that's Kovacs indole. There's a Kovacs oxidase test. So you perform it the same way using CTA or filter paper. Put organism on the filter paper and then add a drop of the reagent. Again, they have these pinch vials. Inside it has a glass vial. You pinch it, break the glass, and you're not going to cut yourself. Don't worry. And you squeeze out a drop or two onto a swab, and then you look for color reaction 30 seconds later. So hopefully we'll get to do that test either Thursday or Friday. Okay, it's a really, really easy test to perform. Positive is blue to purple color within 10 seconds right away. QC is Pseudomonas aeruginosa, and a negative oxidase test would be any Enterobacteriaceae. It could be E. coli, Enterobacter, Kleb, Serratia. All of those are oxidase negative. Okay, and that's a positive right there with a purple spot, and negative is no purple. Okay, I mentioned the MIO tube, a Motility-Indole-Ornithine. So this test will only do one decarboxylase test, which is the ornithine decarboxylase. I showed you the motility where there's a positive motility where you have diffuse growth throughout the tube, and then the indole test using the dimethylaminocinnamaldehyde. So it tests for motility, indole, and ornithine. This is called combination media because you can do a combination of tests. It's semi-solid media. The substrate includes glucose and the amino acid ornithine for decarboxylation. Okay, there's also a pH indicator from cresol purple and cresol red. And also there's peptone and enrichment. And for the indole, we're looking for tryptophan. Okay, I mean we're not looking for tryptophan. We're looking for the end product indole from the hydrolyte from tryptophan hydrolyzation. Okay, tryptophan is the substrate. We're looking for the breakdown, and we're looking for the production of indole. So make sure you associate tryptophan and indole. So you inoculate the media, you stab it, goes straight in, goes and pull out straight. One stab in, and you don't use a loop. There's the needles, in the laboratory. Collect a single colony by needle, stab it, and then pull out the same direction. Don't wobble around. Then you incubate it overnight at 37 degrees. Of course, it's a screw cap tube, and as with all screw cap tubes, you want to incubate it with the cap fastened loosely. Motility, like I said, is diffuse growth throughout the tube. A negative is you see the stab line intact. For the decarboxylase test, you look at the lower 1/3 of the tube and if it's positive, it's purple. If it's negative, it's yellow. And the indole tube is a test that's performed last because you're adding the liquid reagent to the top of the tube. And if you see a red or pink color, then that's positive, just like what you saw in the earlier slide. And if it's negative, you got no pink at all. Okay. QC for motility. E. coli is a positive has positive motility. Remember, all Enterobacteriaceae have, if they're motile, not all Enterobacteriaceae, but all motile Enterobacteriaceae is have peritrichous flagella. Club-pneumo, which is Enterobacteriaceae, is negative motility, and that's used for negative control. For ornithine decarboxylase, positive control is Enterobacter cloacae, and negative, again, is Club-pneumo. Indole test, positive is E. coli, negative is Club-pneumo, and you want to perform the indole test last. Okay. Hopefully, when the MIO tubes come in, we can run this test. It's an easy test to do. You stab it, you take your needle, collect some organisms, stab the tube, and incubate it, and then look at the reactions the next day. Pretty easy test to do. Methyl Red Voges-Proskauer, there isn't a tube that we can use to observe this reaction, but if we're going to be doing the API strips that does have Methyl Red and Voges-Proskauer wells, copules that we can see the reaction, the Methyl Red portion of this test will we're looking for the production of enzymes in the mixed acid pathway and the end products of glucose metabolism. And what we're looking for is a red color after we add the methyl red reagent. For the Voges-Proskauer, the two end products are acetone and butanediol. Make sure you know that. Okay, I think I may be asking you the end products of the Voges-Proskauer test, which is acetone and butanediol. I hope you're making little asterisks on these as I'm lecturing, as I tell you all these hints to make sure you know them. And for the Voges-Proskauer test, we're looking for the development of pink or red color when combined with the alpha-naphthol reagent. MRVP broth we're not going to have, and it has these constituents, methyl red, and then ethyl alcohol, distilled water. What you do is you divide the tubes into two, one for the methyl red test and the other one for the Voges-Proskauer test. You inoculate the two broths overnight, and at 37 degrees, add 5 drops of methyl red reagent for the methyl red test, and you're looking for a bright red color for positive. And if you don't get the bright red for the methyl red test, then it's negative. Orange doesn't count, and yellow is definitely negative. E. coli is methyl red positive, and E. cloacae is methyl red negative. Okay, I wish we could get some of these tubes. I'm just having trouble getting regular conventional median supplies. So maybe if we do get it later on this year, we can do it. Or we can do the API strip and you can see the methyl red test and the Voges-Proskauer test. So on the second tube that you split 2.5 mLs, you're going to add Barrett's reagents, which consist of solution A and solution B. So to the tube, the second tube, you're going to add 6 drops. That's what GTTS stands for, 6 drops. I don't know what the direct translation of GTTS, but scientists in the old days And rather than saying drops, I think it's easier to understand drops, but they wrote GTTS, some kind of Latin term. So 6 drops of solution A, which is alpha naphthol, and 2 drops of reagent B, which is 40% potassium hydroxide. Okay, shake the tube, mix the tube a little bit, incubate at room temperature for about 5 minutes, and then look for color change. E. coli is positive, E. coli is negative. So again, you're looking for a red color for positive, okay? Red color for positive on the VP. ONPG stands for orthonitrophenylgalactosidase. Orthonitrophenylgalactosidase. So what you're looking for is the production of beta-galactosidase. So make sure you know, just like tryptophan and indole, for ONPG, associate beta-galactosidase. Okay, the production of beta-galactosidase, that's ONPG. So it's an enzymatic test, and it's related to lactose utilization. So lactose utilization, okay? Organisms testing the organism's ability to ferment lactose at a slow rate. Active beta-galactosidase results in metabolite orthonitrophenol. Okay, this is the rapid test, 0.2 mL of sterile normal saline, inoculate with a loop, and then there's an ONPG disc. So again, this test is on the API strip, and what you're doing, you incubate it for 4 hours at 37 degrees, and you look for a yellow color. On the API strip, it's a very, very faint yellow. Almost a lot of the organisms are, if you get that, not a lot, but E. coli especially, it's pretty common for E. coli, is that the first reaction well is the ONPG. You look at that, if you see a slight tinge of yellow, it's very, very faint, then that's almost a shoo-in for E. coli, that you probably have E. coli. Negative control is Salmonella typhimurium, it's ONPG negative. But E. coli is ONPG positive, yellow reaction. Nitrate reduction, as you recall, all Enterobacteriaceae reduced have the capability of reducing nitrate to nitrite. Okay, nitrate will go to nitrite. Okay, all Enterobacteriaceae will do that. We're looking for the reduction of nitrate to nitrite. So the nitrate media, hopefully we're going to have in the laboratory with a Durham tube, and the Durham tube is to observe gas production. What it is, it's like a small test tube within a tube. It's inverted, so if there's gas production, the gas will collect in the tube, and you can actually see a bubble. So hopefully we can get the nitrate tubes with nitrate media tubes with a Durham tube in it. Okay, that tube, which is inverted, the purpose is to capture gas. So you take your organism, inoculate your liquid media, nitrate tube, incubate it 37 degrees overnight usually, but you can incubate it up to 48 hours. Okay, the reagents are alpha-naphthylamine for reagent A and sulfonilic acid for reagent B. So 2 reagents for the nitrate reduction as well. And then there's zinc dust that is used for further reduction to nitrate. It's almost similar to like the coagulase test. The coagulase test, when you do the coagulase test, you do a slide coagulase first. And if it's negative, if it's positive, you definitely have Staph aureus. Okay, you don't have to proceed. But if you have a negative slide coagulase test, you need to continue on and do a tube coagulase. So it's possible to have a negative coagulase on the slide, and then when you do the tube coagulase, it's possible that you can have a positive tube coagulase, because you can have free coagulase and not have bound coagulase. The tube will measure free coagulase, and the slide will measure bound coagulase. So a couple people got that wrong on the quiz, and that's one of the things I was saying about. I was trying to drive that point home, but apparently it wasn't picked up. Could have been my fault in explanation, and I'll accept that. But for the nitrate reaction, okay, when you do the nitrate test, you're going to inoculate 5 drops of reagents, reagent A and reagent B. Okay, so this is 24 hours later. You take a look at the tube, and you've added your reagents. Okay, you added your reagents. If it turns red within 30 seconds, you add reagent A and reagent B, and it turns red. That's a positive. That's a positive for nitrate. Or remember, all Enterobacteriaceae are nitrate positive. Negative is no color change. Now, if you have a negative color change on this first reaction after adding reagent A and reagent B, you got no color change, then what you do is that no color change, that tube, now you have to add zinc dust. If it remains no color change, then it's positive. Okay, it's a little opposite. And if you get a red after you add the zinc dust and it's red, then that confirms that it's truly negative. So a red color confirms a negative test using zinc dust. If you add zinc dust and there's no color change, it's still clear, then that's positive. Okay. I know that's a little confusing. Let me repeat that. Okay, so you're gonna do the nitrate test. You're gonna add reagent A, reagent B. If you get the red color, you need to stop. It's already positive. Nitrate has been reduced to nitrite. Okay. So, but if you don't get a red color, you got a negative color change. It's still positive that nitrate has reduced all the way to, instead of nitrite, to N2. Okay. Nitrogen gas. So we want to detect if the nitrate has reduced all the way past the nitrite, all the way to N2. Okay, so if it has, then when you get this negative color, you're not going to get the nitrate. That means it's it's nitrite. Negative color is nitrite negative. That's what you're testing for. But it could have gone all the way to N2. So when you add the zinc dust, a no color change confirms that it's positive. Add no color change is positive. If it's truly negative and it did not go to N2, then that's when zinc dust will give you a red color. I'll mention that again tomorrow, and then again when we do the review for Enterobacteriaceae. But it's similar to the coagulase test. A negative reaction, you have to continue on. And in this case, we add zinc dust. A red color is truly negative. Okay, a red color with zinc is truly negative. In QC, E. coli is positive for nitrate reduction, and anything that's not in the Enterobacteriaceae is a good QC for nitrate reduction. It's not in Enterobacteriaceae, it's a good negative control for nitrate reduction. Here's the API strips. I wish we can do these. I think we will. If they're in the laboratory, we're gonna make some 0.5 McFarland standard suspension, inoculate the tubes, and then add the reagents, and then we'll take a look at the color reactions. If we're not going to identify the organism, at least we'll take a look at the color reactions. Okay, that's the API strip where you actually generate a 7-digit code, and the 7-digit code that we generate I can give you the procedure for doing it, will correspond to a particular organism, and it'll tell you from the database your percent confidence on whether or not that's E. coli, E. coli, Club pneumo, etc. So you have to have like greater than 99% confidence or 95%. It'll tell you the confidence from the program that you enter the 7-digit code in. So this is a pretty good system. It can be a little bit expensive, but it's a pretty good identification system for The E stands for Enterobacteriaceae. There's the API 20NF for the non-fermenters. So if I use the term non-fermenter, that's non-glucose fermenter, like after Enterobacteriaceae, things like the Aeromonas, the Plesiomonas, those are all non-fermenters. They don't ferment glucose. So that group is called well, the collective term is they're non-fermenters, but there's aerobic GNRs and facultative GNRs. Those are not members of Enterobacteriaceae, but don't get that mixed up with non-lactose fermenters, because those are specific for lactose fermentation. There's non-fermenters and non-lactose fermenters. I don't want you to be confused. If the term is non-lactose fermenters, we're still talking about Enterobacteriaceae, okay? Okay, the Proteus group. Proteus vulgaris, Proteus mirabilis, and Proteus penneri. The two main ones are vulgaris and mirabilis. Those are the most frequently isolated. Mirabilis is the most common one, followed by vulgaris. Clinical significance, a lot of times I see it in junk, but it can be significant for urinary tract infection, wound infection, and nosocomial infections. The handout I'm talking about here is the Ewing's flowchart. I'm going to talk about the Ewing's flowchart later on too, and especially when I do the review tomorrow. Okay, one colony here, and from this colony, a lot of swarming took place. Those are swarms. These waves are swarms from this one colony here. So Proteus, that's the main characteristic of Proteus. It's a swarmer. It has a particular foul smelling smell. These colors are colorless, and here's that term, non-lactose fermenter, as opposed to non-fermenter. A non-fermenter, remember, it's not Enterobacteriaceae, but a non-lactose fermenter is colorless when on McConkey. So you can see whether or not an organism is a lactose fermenter or a non-lactose fermenter by looking at the McConkey plate. If it's a lactose fermenter, it's going to be pink. If it's a non-lactose fermenter, it's going to be colorless. Now, when you're doing workups for pathogens, okay, you need to be working up the non-lactose fermenters, because the main pathogens, Salmonella and Shigella, are non-lactose fermenters. They produce colorless colonies on McConkey. So anytime you see colorless colonies on McConkey, that's what you go after, that's what you identify, and that's what you work up for sensitivities, okay? On H&E, the colonies are colorless also without black center H2S. Now, Salmonella is colorless, but Salmonella is H2S positive. So the colonies of Salmonella on an H&E plate, H&E is the hectoin enteric agar, and like I said, it's colorless. For Proteus, it's colorless with no black center. XLD we won't use, but that's yellow to colorless. Indole differentiates, like I mentioned earlier, differentiates Proteus mirabilis. It's negative for mirabilis, and it's differentiated from mirabilis with Proteus vulgaris, which is indole positive. Remember the three indole positive organisms: E. coli, Proteus vulgaris, and Klebsiella oxytocca. Proteus species, 3 colonies, and swarming effect from those 3 colonies, okay? Swarmer. Providencia, 2 main species, Providencia stewartii and Providencia rectori, clinically significant on a urine culture causing urinary tract infection and present in nosocomial infection and wound infection. So Providencia can be important, like I said, in UTI, stewartii and rectori. The handout again is the flow chart. So on blood, Providencia is a non-swarmer. It's just a regular gray colony. But on McConkey, the problem is it's a non-lactose fermenter, so it's colorless. Even though Providencia may not be a significant pathogen, but it can be pathogenic, it can be clinically significant, you have to work it up. because it's a non-lactose, it's a colorless clear colony on MacConkey. Remember, the two morphologies on MacConkey are colorless and clear. And also too with Proteus, Proteus swarms on blood and TSA, but it will not swarm on MacConkey. Okay, Proteus will not swarm on MacConkey. But it will be a non-lactose fermenter colorless. That's Providencia. H&E is colorless, XOD is yellow and colorless. So when working the microbiology bents, you always look on MacConkey to see if you have any colorless colonies, because those are the colonies that you have to work up. Morganella is also in the same group. Morganella has a similar clinical significance to Providencia, UTI, wound infections, but meningitis. So Morganella can be a pretty significant pathogen. Usually, if there's 2 or 3 colonies on a primary streak of this organism, it's probably not clinically significant. But when you get colonies in the 3rd and 4th quadrant, you know, when you streak, that at that quantity, that means it's clinically significant, that you can probably say 3 or 4 plus present. Okay, that's how you measure quantity. If it's only in the primary streak, it's probably 1 plus. The secondary streak 1 to 2 plus. The 3rd and 4th, I like to go 4. That's 3 or 4 plus. And colonies that far out are clinically significant. Okay, Morganella, handout again, take a look at it. It's a non-swarmer. It's in the Proteus group. Again, it's colorless, non-lactose fermenter. So this one definitely has to work up. You have to be suspicious that any colorless colony on MacConkey could be a Salmonella or a Shigella. The Salmonella or Shigella is reportable to public health. Okay, H&E is colorless, XOD is red or colorless. Okay, and that's it for Enterobacteriaceae 7, our lecture 7. Are there any questions? Are there any questions? We're going to go over this, especially when I go over the review and when I go over the Ewing's charts. Okay, the Ewing's charts are divided nicely, and you're going to be tested on this Ewing's charts. But don't be intimidated. Don't be intimidated by the Ewing's charts, and I'll make it easy for you to learn it. Okay. So that is lecture 7.